Hello, I'm Yasaman Torabi, a PhD candidate of Department of Electrical and Computer Engineering at McMaster University. Today I want to talk about large language models and non-negative matrix factorization for bioacoustic signal decomposition. So today I'm going to briefly talk about what is a non-negative matrix factorization and what is a large language model and why we combine them. Then I'll talk about the application of this algorithm in biomedical sound separation specifically, and a brief review and visualization of our results. Then I proceed to the advantages of using LLM as well as their limitation and future work. So if I want to start, I want to talk first about the motivation of why we are doing this. So we want to do BSS, or blind source separation. Blind source separation is the problem of recovering source signals from a mixture with limited information or no information about the sources and the mixing process. So in here, you're specifically working with hard and long sounds, and these hard and long sounds are gonna often overlap when you are recording them in auscultation. And the manual separation of them is really hard, and it might have a lot of errors. So our goal is to separate the sources with mathematical algorithms, and we pick matrix factorization as a good option. And then later, we use LLMs to improve this source separation beyond the typical NMF and matrix factorizations. And what is non-negative matrix factorization? So non-negative matrix factorization is basically a linear decomposition method that factorizes a non-negative matrix that we have into two lower rank non-negative matrices. So if we name it as V, it's going to be separated to W and H. One of them is the observed mixed signal, which is our input. And then we have W and H. One of them is the basis, the other one is the activation. So when we find the W and H, we have the hard and long sound, and how each of them contribute to this mixture, okay? So one of them is going to be the activation, how important they are, which are the H, and then W's, which are the bases, which is going to be, one of them is going to be the hard, one of them is going to be the long sound. And this method decomposes the signals into interpretable and non-overlapping components, which is really useful for unsupervised source separation in bio-acoustic signals or different acoustic signal separations. And now, what is a large language model, or LLM? So LLM is typically an AI-powered deep neural network which is trained on big text data to understand and generate human languages, and it can learn contextual relationship between the words, phrases, and concepts. The good thing about the LLM is that they can reason and classify or extract semantic insights. So in this study, we're using LLM to provide insight for disease prediction based on the sounds that we separated from our mixture. So why did we combine NMF and LLM? So NMF part is breaking out the mixed signal into meaningful patterns and separate it, and then let LLM bring reasoning and language level insight on that. So this combination supports more adaptive signal analysis so that we can enable context-aware biomedical sound separation. So it can help us to have a better and explainable algorithm. So compared to standard NMF, this combination can have a kind of intelligent guidance, AI-based guidance, and it is adaptive via feedback that we get from the LLM. So it is informed by learned patterns, and it is not only reliable to relied on mathematical things that we have in a standard NMF. It has mathematical parts as well as explainable AI as well as semantic interpretation. And how our algorithm is used in biomedical sound separation. So our input is in this project is gonna be the mixed time frequency features that we got from the hard and long sound. We recorded this hard and long sound from clinical mannequins using digital stethoscope. And our output is gonna be clean hard sound and clean long sound. So the NMF is separating the signals based on the patterns, and LLM is guiding this separation and kind of help it to explain it using the learned knowledge. And here are some of the qualitative results that we get. This is the visualization of the time frequency domain visualization of the waveforms, and the time frequency spectrograms and the waveforms. This is a spectrogram and this is the waveform. Illustrate how from this mixture the algorithm is effectively separating the acoustic sources. One of them is the long sound, the other one is the hard sound. And the LLM is enhancing this separation by linking some pattern to potential disease. So in here we have the initial mix sound, which is an overlapping time frequency pattern that is going to be really hard to diagnose directly what's happening. But in here we have long sound, and here another hard sound that's separated from this mixture. In the long sound we can see, for example, in here we can see some low frequency bursts, which is typically matched to wheezing. So the LLM is suggesting possible respiratory abnormality in here, such as wheezes. And for the hard sound, we have some regular peaks that they are disrupted by some irregular intervals. And the LLM is suggesting potential presence of atrial fibrillation in here. So the advantage of using LLM in this project is that the LLM can understand complex biomedical context, and it can learn from large-scale medical data. This LLM can enhance signal separation by recognizing the patterns and add some semantic insight to the raw signal features. So we're going to have some text-based explanation on what we have in the separated features. So it enables interpretable results and explainable AI, and supports a smarter decision making in diagnostics. So in future work, we're gonna work on improving the efficiency of it and training it on real clinical data of real people, real human. And one of the limitations that we need to consider when we are using NMF is that NMF can work only on the negative signals, and it has some limitation in handling nonlinear or noisy signals. And about the LLMs are really high computational costs, so we need to kind of work to find algorithms that need less computational costs. And these things, especially because of the potential diagnosis errors, are still in early stages, and they need more validation if we want to use them in real scenarios. So in summary, in this project, we are using NMF and LLM together to separate hard and long sound. And the negative matrix factorization is separating the acoustic signals. And the large language model is enhancing the interpretation of the separated sound. And if you look at a spectrogram and the waveforms of the separated sound, we can confirm clear separation of the hard and long components from the initial mixture. And combining these techniques lead to more accurate and clinical meaningful sound separation in future. So here are some references, some of the preprints that we have and some of the papers that we had that we used for making these things that I just talked about in this presentation. And thank you very much. In case you have any questions or suggestions on this talk, feel free to drop me a line.